Hello, welcome to World of Microbes. In our genetic series so far, we have unlocked the secrets of the cellular control center. First, we watched the cell meticulously duplicate its entire genome so it could divide and multiply, a process called DNA replication. In our last video, we watched the cell open up its master blueprint, DNA, and copy a single gene into a mobile, single-stranded message. That process was transcription, and it left us with a brand new molecule of messenger RNA, or mRNA, floating out of the nucleus. But a recipe on a piece of paper doesn't feed anyone. To actually build a living organism, that message has to be transformed into a functional, physical structure. Welcome to part 2 of the central dogma, translation. Today, we are going to look at the master workforce making this happen, RNA. Then, we will watch it build a protein from scratch, step by step. Think of your DNA as the ultimate biological hard drive. It sits safely inside the nucleus of your cells, holding the master code for everything you are. But a hard drive is useless without software to run it, or a factory to build what the code dictates. Enter RNA. For decades, textbooks treated RNA like a basic errand boy, a simple middleman that takes instructions from DNA and delivers them to the cell. But we were dead wrong. RNA is the builder, the manager, the assassin, and the engine of evolution. Without it, DNA is just an unread book. Today, we break down why RNA is the most versatile molecule on Earth, how it builds life from scratch, and how it's revolutionizing modern medicine. Let's get into it. To understand RNA, we first have to stack it up against its famous sibling, DNA. They look similar, but their structural differences change everything. First, structure. DNA is a double helix, two strands twisted together like a secure, stable ladder. RNA is typically a single strand, making it highly flexible, dynamic, and capable of folding into complex 3D shapes. Second, the sugar backbone. DNA uses deoxyribose, while RNA uses ribose. That deoxys means DNA lacks one oxygen atom. By keeping that oxygen atom, RNA becomes chemically more reactive, ideal for a molecule that needs to do active work, not just sit in storage. Finally, the code itself. DNA uses for chemical bases, adenine, thymine, cytosine, and guanine. RNA uses 3 of the same, but swaps out thymine for uracil. Why uracil? It takes less energy for a cell to produce, which is perfect for RNA because the cell needs to mass produce it constantly. Inside your cells, a massive manufacturing operation is happening right now, managed by 3 main types of RNA. Think of it as a busy construction site. The 3 primary types of RNA are messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, rRNA. Together, they work as a coordinated system to translate genetic information from DNA into functional proteins within your cells. First up is mRNA, or messenger RNA. When a cell needs to make a protein, say, insulin or collagen, it doesn't unlock the master DNA blueprint. Instead, it creates a temporary photocopy of that specific gene. This is mRNA. mRNA, messenger RNA, is a genetic molecule that acts as a blueprint, carrying instructions from your DNA to the cell's protein-making factories, ribosomes. It tells your body exactly which proteins to build to function correctly. It carries the code out of the vault and down to the factory floor. Waiting on the factory floor is rRNA, or ribosomal RNA. Ribosomes are the cellular machines that build proteins, and they are actually constructed out of rRNA and proteins mixed together. The rRNA forms the physical structure and the catalytic engine that links materials together. It accounts for roughly 80% of all RNA in a typical cell and acts as the mechanic that translates genetic codes into proteins. Finally, we have tRNA, or transfer RNA. If mRNA is the blueprint and rRNA is the factory machine, tRNAs are the delivery trucks. Each tRNA molecule carries one specific amino acid, the building blocks of proteins. It reads the mRNA blueprint 3 letters at a time, making sure it delivers the exact right amino acid to the assembly line. Let's look at how this happens step by step. The first major phase of life is transcription, which means to write out. Inside the nucleus, an enzyme called RNA polymerase latches onto your DNA. It unzips the double helix, exposing the gene needed for the job. The enzyme then reads the DNA template and builds a matching strand of mRNA. Think of it like matching puzzle pieces. Wherever the DNA has a guanine, the enzyme adds a cytosine. Wherever the DNA has an adenine, the enzyme attaches a uracil. Once the gene is fully copied, the freshly minted mRNA strand detaches, the DNA zips back up perfectly unharmed, and the mRNA exits the nucleus into the cell's vast cytoplasm. It's a flawless, non-destructive copying process. Now comes the magic: translation. This is where the digital code of nucleic acids turns into the physical reality of a protein. The mRNA strand binds to a ribosome. The ribosome reads the mRNA strand in 3-letter increments called codons. Each codon is a specific instruction for an amino acid. For example, the codon AUG is the universal start signal. A tRNA molecule with a matching 3-letter key, called an anticodon, docks into the ribosome, bringing its specific amino acid cargo. The ribosome then clicks over to the next codon, a second tRNA arrives, and the ribosome welds their amino acids together with a peptide bond. This repeats hundreds of times, a hyperfast molecular sewing machine, building a chain of amino acids. Once a stop codon is reached, the chain is released, instantly folding into a highly specific 3D shape. Congratulations, you just built a functional protein. For decades, scientists believed that mRNA, tRNA, and RNA were the only variants that mattered. They noticed that only about 2% of human DNA actually codes for proteins. They called the rest junk DNA. But they were completely wrong. We now know that a massive chunk of that remaining DNA is used to build non-coding RNAs. These are the middle managers of your genome. Molecules like microRNA, mRNA, and small interfering RNA, siRNA, act like cellular dimming switches or corporate hitmen. If a cell produces too much of a certain mRNA blueprint, these tiny non-coding RNAs will hunt them down, bind to them, and chop them up before they can reach a ribosome. They regulate gene expression, dictate cell specialization, and protect you from viruses. They are the software code that controls the entire cell. Because we finally understand RNA, we are entering a golden age of medicine. In the past, creating a vaccine or drug meant painstakingly growing proteins in a lab and injecting them into patients. With mRNA technology, we don't have to do that anymore. We can simply write the genetic code for the cure, put it in a microscopic delivery bubble, and inject it. Your own cellular machinery reads the blueprint and builds the medicine for you. This technology is already being deployed to create personalized cancer vaccines, reverse autoimmune disorders, and power gene editing tools like CRISPR. RNA isn't just a helper molecule, it is the fundamental bridge between the digital code of life and the physical world. As we discovered that RNA is the ultimate engine of life, the software that makes our biological hardware run. But how exactly does the cell create RNA in the first place? How do you take a massive, tightly locked library of DNA and perfectly photocopy just one specific recipe without making a single mistake? The answer is transcription. It is the absolute first step in gene expression, happening millions of times inside you right now. If this process fails by even a single letter, it can cause everything from genetic disorders to cancer. But when it works, it is a masterpiece of molecular engineering. Today, we are zooming deep inside the nucleus to watch this microscopic photocopy machine in action, step by step. Let's roll. To make an RNA molecule, the cell relies on a single master enzyme. Meet RNA polymerase. If DNA is a locked book, RNA polymerase is the reader, the typewriter, and the editor all wrapped into one. Its job sounds simple: find a gene on the DNA strand, open it up, and make a complementary RNA copy. But think about the scale of this task. The human genome contains over 3 billion base pairs of DNA. If you printed out your DNA sequence, it would fill a library of books. RNA polymerase has to navigate through that massive wilderness of data, find the exact line of text it needs, and ignore everything else. How does it know where to start, where to stop, and how to read the code? It all comes down to 3 distinct chemical phases: initiation, elongation, and termination. Like translating a book from one language into another, the codons on a strand of mRNA must be translated into the amino acid alphabet of proteins. Translation is the process of synthesizing a chain of amino acids called a polypeptide. Translation requires 2 major aids: first, a translator, the molecule that will conduct the translation, and second, a substrate on which the mRNA strand is translated into a new protein, like the translator's desk. Both of these requirements are fulfilled by other types of RNA. The substrate on which translation takes place is the ribosome. Remember that many of a cell's ribosomes are found associated with the rough ER, and carry out the synthesis of proteins destined for the Golgi apparatus. Ribosomal RNA, rRNA, is a type of RNA that, together with proteins, composes the structure of the ribosome. Ribosomes exist in the cytoplasm as 2 distinct components, a small and a large subunit. When an mRNA molecule is ready to be translated, the 2 subunits come together and attach to the mRNA. The ribosome provides a substrate for translation, bringing together and aligning the mRNA molecule with the molecular translators that must decipher its code. The other major requirement for protein synthesis is the translator molecules that physically read the mRNA codons. Transfer RNA, tRNA, is a type of RNA that ferries the appropriate corresponding amino acids to the ribosome, and attaches each new amino acid to the last, building the polypeptide chain one by one. Thus tRNA transfers specific amino acids from the cytoplasm to a growing polypeptide. The tRNA molecules must be able to recognize the codons on mRNA and match them with the correct amino acid. The tRNA is modified for this function. On one end of its structure is a binding site for a specific amino acid. On the other end is a base sequence that matches the codon specifying its particular amino acid. This sequence of 3 bases on the tRNA molecule is called an anticodon. For example, a tRNA responsible for shuttling the amino acid glycine contains a binding site for glycine on one end. On the other end it contains an anticodon that complements the glycine codon. GGA is a codon for glycine, and so the tRNA's anticodon would read CCU. Equipped with its particular cargo and matching anticodon, a tRNA molecule can read its recognized mRNA codon and bring the corresponding amino acid to the growing chain. Translation involves translating the sequence of a messenger RNA, mRNA, a molecule to a sequence of amino acids during protein synthesis. It is the process in which ribosomes in the cytoplasm or ER synthesize proteins after the process of transcription of DNA to RNA. Ribosomes exist normally as separate subunits that are composed of protein and rRNA. The subunits come together to form a ribosome when they bind to an mRNA near its 5' end. On binding to an mRNA, the ribosome reads the nucleotide sequence from the 5' to 3' direction, synthesizing the corresponding protein from amino acids in an N-terminal, amino terminal, to C-terminal, carboxyl terminal, a direction. Ribosomes are located in the cytosol, either freely floating or associated with the endoplasmic reticulum. They serve to synthesize proteins. Each prokaryotic ribosome, shown schematically, has 3 binding sites for tRNAs. The aminoacyl tRNA binding site, or a site, is where, during elongation, the incoming aminoacyl tRNA binds. The peptidyl tRNA binding site, or P site, is where the tRNA linked to the growing polypeptide chain is bound. The exit site, or E site, is a binding site for tRNA following its role in translation and prior to its release from the ribosome. All 3 sites, A, P and E, are formed by the rRNA molecules in the ribosome. Protein synthesis, or translation, takes place in 3 stages: initiation, elongation and termination. During initiation, the mRNA ribosome complex is formed and the first codon, always August, binds the first amino acid RNA, called initiator tRNA. During the elongation phase, the other codons are read sequentially and the polypeptide grows by addition of amino acids to its C-terminal end. This process continues until a termination codon, stop codon, which does not have a corresponding amino acid tRNA with which to base pair, is reached. At this point, protein synthesis ceases, termination phase, and the finished polypeptide is released from the ribosome. Synthesis of amino acid tRNAs is crucially important for two reasons. Each amino acid must be covalently linked to a tRNA molecule in order to take part in protein synthesis, which depends upon the adapter function of tRNA to ensure that the correct amino acids are incorporated. The covalent bond that is formed between the amino acid and the tRNA is a high-energy bond that enables the amino acid to react with the end of the growing polypeptide chain to form a new peptide bond. For this reason, the synthesis of amino acid tRNA is also referred to as amino acid activation. Each tRNA molecule has a cloverleaf secondary structure with the anticodon accessible at the end of the anticodon stem loop. During synthesis of the amino acid tRNA, the amino acid is covalently bound to the A residue of the CCA sequence at the 3' end. Each tRNA molecule carries only a single amino acid. The attachment of an amino acid to a tRNA is catalyzed by an enzyme called amino acid tRNA synthetase. A separate amino acid tRNA synthetase exists for every amino acid, making 20 synthetases in total. The synthesis reaction occurs in two steps. The first step is the reaction of an amino acid and ATP to form an amino acid adenylate, also known as amino acid AMP. In the second step, without leaving the enzyme, the amino acid group of amino acid AMP is transferred to the 3' end of the tRNA molecule to form amino acid tRNA. Initiation begins with the binding of IF1 and IF3 to the small 30S ribosomal subunit. Their role is to stop the 30S subunit binding to the 50S subunit in the absence of mRNA and FMET tRNA FMET, which would result in a non-functional ribosome. The small subunit then binds to the mRNA via the Shine-Adalgano sequence and moves 3' along the mRNA until it locates the August initiation codon. The initiator tRNA charged with N-formal methionine and in a complex with IF2 and GTP now binds. IF3 is released. The complex of mRNA, FMET tRNA FMET, IF1, IF2 and the 30S ribosomal subunit is called the 30S initiation complex. The large, 50S, ribosomal subunit now binds, with the release of IF1 and IF2 and hydrolysis of GTP, to form a 70S initiation complex. At the start of the first round of elongation, the initiation codon, August, is positioned in the P site with FMET-TRAFMET bound to it via codon-anticodon base pairing. The next codon in the mRNA is positioned in the A site. Elongation of the polypeptide chain occurs in 3 steps called the elongation cycle, namely amino acid tRNA binding, peptide bond formation and translocation. Amino acid tRNA binding. The corresponding amino acid tRNA for the second codon binds to the A site via codon-anticodon interaction. Binding of the amino acid tRNA requires elongation factor EF2 and GTP, which bind as an amino acid tRNA, EF2-GTP complex. Following binding, the GTP is hydrolyzed and the EF2 is released, now bound to GDP. Before the EF2 molecule can catalyze the binding of another charged tRNA to the ribosome, it must be regenerated by a process involving another elongation factor, EFTS. This regeneration is called the EF2-EFTS exchange cycle. First, EFTS binds to EF2 and displaces the GDP. Then GTP binds to the EF2 and displaces EFTS. The EF2-GTP is now ready to take part in another round of elongation. Peptide bond formation. The second step, peptide bond formation, is catalyzed by peptidyl transferase. In this reaction, the carboxyl end of the amino acid bound to the tRNA in the P site is uncoupled from the tRNA and becomes joined by a peptide bond to the amino group of the amino acid linked to the tRNA in the A site. Translocation. In the third step, a complex of elongation factor EFG, also called translocase, and GTP, i.e. EFG or GTP, binds to the ribosome. Three concerted movements now occur, collectively called translocation. The deacylated tRNA moves from the P site to the E site. The dipeptidyl tRNA in the A site moves to the P site, and the ribosome moves along the mRNA, 5' to 3' by 3 nucleotides to place the next codon in the A site. During the translocation events, GTP is hydrolyzed to GDP and inorganic phosphate, and EFG is released ready to bind more GTP for another round of elongation. After translocation, the A site is empty and ready to receive the next amino acid RNA. The A site and the E site cannot be occupied simultaneously. Thus the deacylated tRNA is released from the E site before the next amino acid tRNA binds to the A site to start a new round of elongation. Elongation continues, adding one amino acid to the C-terminal end of the growing polypeptide for each codon that is read, with the peptidyl tRNA moving back and forth from the P site to the A site as it grows. Eventually, one of 3 termination codons, also called stop codons, becomes positioned in the A site. These are UAG, UAA, and UGA. Unlike other codons, prokaryotic cells do not contain amino acid tRNAs complementary to stop codons. Instead, one of 2 release factors, RF1 and RF2, binds instead. RF1 recognizes UAA and UAG, whereas RF2 recognizes UAA and UGA. A third release factor, RF3, is also needed to assist RF1 or RF2 interaction with the ribosome. Thus either RF1 plus RF3 or RF2 plus RF3 bind depending on the exact termination codon in the A site. RF1 or RF2 binds at or near the A site, whereas RF3 or GTP binds elsewhere on the ribosome. The release factors cause the peptidyl transferase activity to transfer the polypeptide to a water molecule instead of to amino acid tRNA, effectively cleaving the bond between the polypeptide and tRNA in the P site. The free polypeptide now leaves the ribosome, followed by the mRNA and free tRNA, and the ribosome dissociates into 30S and 50S subunits ready to start translation again. Protein synthesis involves translating the sequence of a messenger RNA, mRNA, a molecule to a sequence of amino acids during protein synthesis. It is the process in which ribosomes in the cytoplasm or ER synthesize proteins after the process of transcription of DNA to RNA. Ribosomes exist normally as separate subunits that are composed of protein and rRNA. Eukaryotic ribosomes are larger, the 80S, and more complex than prokaryotic ribosomes, 70S. The subunits come together to form a ribosome when they bind to an mRNA near its 5' end. On binding to an mRNA, the ribosome reads the nucleotide sequence from the 5' to 3' direction, synthesizing the corresponding protein from amino acids in an N-terminal, amino terminal, to C-terminal, carboxyl terminal, direction. Ribosomes are located in the cytosol, either freely floating or associated with the endoplasmic reticulum. They serve to synthesize proteins. Each prokaryotic ribosome, shown schematically, has 3 binding sites for tRNAs. The amino acid tRNA binding site, or a site, is where, during elongation, the incoming amino acid tRNA binds. The peptidyl tRNA binding site, or P site, is where the tRNA linked to the growing polypeptide chain is bound. The exit site, or E site, is a binding site for tRNA following its role in translation and prior to its release from the ribosome. All 3 sites, A, P, and E, are formed by the rRNA molecules in the ribosome. The overall mechanism of protein synthesis in eukaryotes is basically the same as in prokaryotes. However, there are some significant differences. Whereas a prokaryotic ribosome has a sedimentation coefficient of the 70S and subunits of 30S and 50S, a eukaryotic ribosome has a sedimentation coefficient of 80S with subunits of 40S and 60S. The composition of eukaryotic ribosomal subunits is also more complex than prokaryotic subunits, but the function of each subunit is essentially the same as in prokaryotes. In eukaryotes, each mRNA is monocistronic, that is, discounting any subsequent post-translational cleavage reactions that may occur, the mRNA encodes a single protein. In prokaryotes, many mRNAs are polycistronic, that is, they encode several proteins. Each coding sequence in a prokaryotic mRNA has its own initiation and termination codons. Initiation of protein synthesis in eukaryotes requires at least 9 distinct eukaryotic initiation factors, EIFs, compared with the 3 initiation factors, EIFs, in prokaryotes. In eukaryotes, the initiating amino acid is methionine, not N-formal methionine as in prokaryotes. As in prokaryotes, a special initiator tRNA is required for initiation and is distinct from the tRNA that recognizes and binds to codons for methionine at internal positions in the mRNA. When charged with methionine ready to begin initiation, this is known as met-tRNA met. The main difference between initiation of translation in prokaryotes and eukaryotes is that in bacteria, a Shine-Dalgarno sequence lies 5' to the August initiation codon and is the binding site for the 30S ribosomal subunit. In contrast, most eukaryotic mRNAs do not contain Shine-Dalgarno sequences. Instead, a 40S ribosomal subunit attaches at the 5' end of the mRNA and moves downstream, i.e. in a 5' to 3' direction, until it finds the August initiation codon. This process is called scanning. Prokaryotic translation requires no helicase, presumably because protein synthesis in bacteria can start even as the mRNA is still being synthesized, whereas, in eukaryotes, transcription in the nucleus and translation in the cytoplasm are separate events that allow time for mRNA secondary structure to form. Protein synthesis, or translation, takes place in 3 stages: initiation, elongation, and termination. The first step is the formation of a pre-initiation complex consisting of the 40S small ribosomal subunit, met-tRNA met, EIF2, and GTP. The pre-initiation complex binds to the 5' end of the eukaryotic mRNA, a step that requires EIF for F, also called cap binding complex, and EIF3. The EIF for F complex consists of EIF for A, EIF for E, and EIF for G. EIF for E binds to the 5' cap on the mRNA, whilst EIF for G interacts with the poly-A binding protein on the poly-A tail. The EIF for A is an ATP-dependent RNA helicase that unwinds any secondary structures in the mRNA, preparing it for translation. The complex then moves along the mRNA in a 5' to 3' direction until it locates the August initiation codon, i.e. scanning. The 5' untranslate regions of eukaryotic mRNAs vary in length but can be several hundred nucleotides long and may contain secondary structures such as hairpin loops. These secondary structures are probably removed by initiation factors of the scanning complex. The initiation codon is usually recognizable because it is often, but not always, contained in a short sequence called the Kozak consensus, 5' ACCAUGG3'. Once the complex is positioned over the initiation codon, the 60S large ribosomal subunit binds to form an 80S initiation complex, a step that requires the hydrolysis of GTP and leads to the release of several initiation factors. Elongation depends on eukaryotic elongation factors. 3 elongation factors, EEF1A, EEFIB, and EEF2, are involved, which have similar functions to their prokaryotic counterparts EF2, EFTS, and EFG. At the end of the initiation step, the mRNA is positioned so that the next codon can be translated during the elongation stage of protein synthesis. The initiator tRNA occupies the P site in the ribosome, and the A site is ready to receive an amino acid tRNA. During chain elongation, each additional amino acid is added to the nascent polypeptide chain in a 3-step microcycle. The steps in this microcycle are: positioning the correct amino acid tRNA in the A site of the ribosome, forming the peptide bond, and shifting the mRNA by 1 codon relative to the ribosome. Although most codons encode the same amino acids in both prokaryotes and eukaryotes, the mRNAs synthesized within the organelles of some eukaryotes use a variant of the genetic code. During elongation in bacteria, the deacylated tRNA in the P site moves to the E site prior to leaving the ribosome. In contrast, although the situation is still not completely clear, in eukaryotes the deacylated tRNA appears to be ejected directly from the ribosome. Termination of elongation depends on eukaryotic release factors. In eukaryotes, eukaryotic release factor ERF1 recognizes all three termination codons, UAA, UAG, and UGA, and, with the help of protein ERF3, terminates translation. Upon termination, the ribosome is disassembled and the completed polypeptide is released. And there you have it! The journey is complete. We started with DNA replication to keep the genetic code alive across generations. We used DNA transcription to draft a mobile copy of a single gene. And today, we watched RNA and translation turn those letters into real, living anatomy. If you missed the first two parts of this incredible molecular journey, make sure to click the on-screen links or check the description to watch our full breakdowns of replication and transcription. Hit that like button if this made biology easier to understand, subscribe for more cellular deep dives, and leave a comment telling me what topic we should tackle next. See you in the next video.